Thanks for joining us. I'm Dr. Alan Brown, and thanks for joining this recording of Lipid Insights. We are recording at the National Lipid Association meeting in Miami, so you may hear some background noise, but it's been a very exciting meeting. And I have the pleasure of talking today with Dr. Rebecca Juliano. She is the Chief Development Officer at Maria Pharmaceuticals.
Therapeutics.
Okay. And gave a very provocative presentation about a new therapy targeting angiopoietin-like protein number 4. We've had a lot of drugs developed that target ANGPTL4, so I'm looking forward to hearing what you found, why you chose that as a target, what you're looking at for outcomes. So thank you very much for joining us.
Thank you for having me. I'm excited to be here.
So tell me a little bit about ANGPTL4 as a target. What I know in the past there were some attempts to develop therapies against it that were kind of abandoned. So how did you get to where you got to now, and what was the basis for you kind of taking up the ANGPTL4 story?
Sure. So the history is, angiopoietin-like 4 was discovered about 25 years ago. It's a potent inhibitor of lipoprotein lipase, and very early the genetics looked compelling that there could be a cardiovascular or even an anti-diabetic impact. There was an early knockout mouse model, and when it was put on a very high saturated fat diet, there was pretty severe adversity. So lipid accumulation in mesenteric lymph nodes that led to localized inflammation, systemic inflammation, and reduced survival. So really, as folks were looking at angiopoietin-like 4 or angiopoietin-like 3 or 8 or even ApoC3 and other targets around lipoprotein lipase, I think folks were basically scared away from it because of the knockout mouse model findings. And so our company was started a few years ago. Our chief scientific officer, Ethan Weiss, was at UCSF. He was very interested in sort of metabolic types of considerations and in genetically validated potential targets. And so he started working on considering what some of these targets could be. And, a few steps later, we ended up in-licensing MAR-001, which is a monoclonal antibody targeted against angiopoietin-like 4. And over the last few years, a lot of the work we've been doing is to address that potential risk. Now, the first step you might do is how can we get over our concern or the concerns that were there for the knockout mouse model? That started with human genetics. We're fortunate that there's very common loss of function mutations, so you can look at this pretty broadly. And then there are some less common variants, but where you get essentially a total knockout. So you can look at people that essentially are knockout, patients. And what we see with all of these folks, first off, is that they live very long, healthy lives, so it seems very safe to have this knocked out, in our gene. There's a substantial reduction in cardiovascular risk, so about a 70% reduction in your risk of cardiovascular disease and about a 50% reduction in your risk of type 2 diabetes. So that was the beginning of thinking that, maybe these mouse model findings were not translatable. And then over the last few years, we've done numerous preclinical studies, including a 9-month toxicology study in monkeys. That got us into our first in-human study, and then we just recently finished our phase 1b/2a study. And in all of those studies, we looked very closely at mesenteric lymph nodes, at lipid accumulation, and a diversity that might come from that, and have really, satisfied ourselves that we don't see this knockout mouse model effect translating into people.
So that's fascinating and obviously encouraging. Since ANGPTL3, 4, and 8 seem to be a factor in where fatty acids are driven based on whether you're in the fed state or you're in the fasting state, do you think the fact that those mice that were fed a high-fat diet had anything to do with the toxicity? And have you looked at what happens with different dietary intake in individuals where you're testing the drug?
Yeah, so in the mouse model, you don't see this unless you have the very high saturated fat. Of course, mice are obligate herbivores, so it's, we think that it has to do with just really pushing this beyond what sort of they were able to tolerate. And in the clinical studies, we actually in our initial studies did limit very high saturated fat diets, or maybe a very high protein diet that could also come with high fat diets. But we didn't see any impact of diet. We did do dietary questionnaires and look to see if we saw any impact or issues. We didn't see any issues, and actually in our next, because of all of the wealth of preclinical and clinical data that we've gathered so far, and in alignment with the FDA, we actually pulled back any dietary restrictions for our upcoming phase 2B study. So I don't think diet is going to be an issue in clinical studies.
Not that it would be bad to recommend a very low-fat diet to everybody.
No, absolutely, and certainly keeping away from saturated fat in general would be better for everyone.
So tell us a little bit about the mechanism of action. I'm sure many of our audience members are not real familiar with ANGPTL4, and none of us are familiar with the mechanism of action of your therapy. So tell us a little bit about, the MOA.
Sure. So maybe I'll back up to lipid metabolism just to start there. So of course, we all know that you can, if you eat, you have chylomicrons produced by your intestines. They have a high triglyceride content on them. Those then are, some of the trigs are pulled off, goes to liver, and then you can produce other trig-rich compound lipoproteins like VLDL, which carries both cholesterol and a lot of triglycerides. Then there are a number of enzymes that can pull triglycerides off of those lipoproteins. Lipoprotein lipase is one of them that's very active. Endothelial lipase is also interesting when you start getting into angiopoietin-like 3. But for ANG4, it is very specifically targeted or focused to lipoprotein lipase. It has tissue expression across the body, but is very heavily expressed in the adipose tissue. And so the idea is that you have angiopoietin-like 4, it inhibits lipoprotein lipase, and so if you are in a state where you don't want to be storing your triglycerides or your fat, such as in a fasted state where you'd want that energy to be going to your muscles, angiopoietin-like 4 basically indirectly, by being at adipose and turning off lipoprotein lipase or inhibiting lipoprotein lipase, targets your tissue, targets your trigs to your muscle. Basically, it's the opposite for angiopoietin-like 3, which has much more skeletal muscle expression, and so therefore would push your storage towards your adipose tissue if you are in a fed state. So these two work in concert, angiopoietin-like 3 and angiopoietin-like 4, to basically direct your triglycerides, depending on your fed or fasted state, to your tissues, either to store or to burn for energy. So again, angiopoietin-like 4 being at the adipose tissue means that it would inhibit you depositing your triglycerides in your adipose. That's actually where you kind of want to put your fat, instead of, say, around your abdomen or around your tissues, or even in skeletal muscle. So the hypothesis here is that if we inhibit angiopoietin-like 4, we allow lipoprotein lipase to do its job. That should primarily be happening at the adipose tissue, which means not only can you pull the trigs out of the plasma, but you're storing them where you want to store them again in your adipose tissue, and particularly in places like gluteofemoral, fat depositions.
So tell us about the targeting then, and what the mechanism of action of the therapy is.
Yeah, so this is a monoclonal antibody, so it's MAR-001. It's a monoclonal antibody against angiopoietin-like 4. We administer it subcutaneously. The 1B2A study that we just finished, it was given every 2 weeks. We are exploring less frequent dosing. We think we have modeling to support that. But basically, it's just an antibody that inhibits angiopoietin-like 4 and essentially deactivates it so that it can no longer inhibit lipoprotein lipase, and then lipoprotein lipase is freed up to do its job.
So tell us a little bit about the results and what you're seeing in the human animal.
Yeah. So, I'm pleased to say that our clinical studies are very similar to our preclinical studies, so that's also always nice to see. Also, again, in the genetics, we can look at lipids and lipoproteins and levels in those folks, and the clinical studies of angiopoietin-like 4 inhibition with MAR-001 are very much mirroring what we see in genetics with people with loss of function. So there's a nice consistency across the data. Overall, we see really substantial reductions in triglycerides and, of course, in remnant cholesterol. Those go hand in hand. Our most recent study, the 1B2A study, enrolled patients with trigs elevated at 150 or higher. And essentially, they were pretty healthy other than that. So they had elevated trigs and remnant cholesterol. They had elevated BMI. Otherwise, we're pretty healthy. We didn't have we had a low prevalence of diabetes, pretty normal HbA1cs, therefore, et cetera. But we saw about a 53% placebo-adjusted reduction in triglycerides, similarly in remnant cholesterol. We saw very nice, about 19% placebo-adjusted increase in HDL cholesterol, and other beneficial effects across the lipid profile. Importantly, we monitored safety through all the standard kind of safety adverse events and safety labs. We didn't see any signals there. We also had a much more detailed look at inflammatory markers because in the mouse model, again, they had localized and then systemic increases in inflammation. We saw no increases in inflammatory markers across the duration of the study and with all doses, so that was very reassuring as well. And then finally, we actually did abdominal MRIs where you could actually count and look at changes in mesenteric lymph nodes and at the fat content of them, and we saw no changes across any of the lymph nodes. So really nice safety findings, and then of course, substantial reductions in lipids that are promising for next stages of development.
That's very encouraging and exciting. It's always cool when a genetic knockout model gives you guidance towards looking for a therapeutic target.
Exactly.
Did you do any testing for insulin sensitivity just in this group, just to get a feel, even though you described a group of patients that didn't sound like they had too much insulin?
Yeah, we did look at it. We saw no changes, but exactly to your point, it's not really the group to see changes in. And so in our next study, we have a phase 2b study we're starting up very soon. We will be looking to get at least 50% of our participants with type 2 diabetes in order to have an opportunity to look at that. We also are going to be running some mechanistic studies, which give us an opportunity to really look at that in much more detail. So.
And how many patients are you hoping to enroll in the phase 2b study?
A little over 200. 216 is the target.
Exciting.
Yeah.
And then do you have long-term goals, obviously, in terms of looking at cardiovascular outcomes?
Absolutely. We hope that this phase 2b study is our pivotal study to take us into a cardiovascular outcome study. So that's exactly what we're looking at doing. Full design still under consideration, but most likely to be a secondary prevention trial. Of course, looking at well-controlled LDL cholesterol and then still having elevated triglycerides and remnant cholesterol.
That is super exciting, and of course we'll be watching this with bated breath because we're always looking for other ways to reduce cardiovascular events. The LDL story is pretty much told, but obviously we have many other interventions now. Let me ask you, if someone were to say, differentiate what you're doing versus targeting ANGPTL3, which you did briefly in the introduction.
Yeah.
How would you describe why you believe there's a significant difference targeting ANGPTL4 versus ANGPTL3 and what the potential benefits might be?
Yeah, I mean, if you just look at where they have impact, so again, ANGPTL4 is very specific to lipoprotein lipase and inhibiting that as opposed to others. And actually, if you look at loss of function of ANGPTL4, gain of function lipoprotein lipase, they look almost identical. So very, very similar there. ANGPTL3 is a little more promiscuous, so it actually impacts both lipoprotein lipase and endothelial lipase. So some of the lipid changes are a little bit different there. And interestingly, when you go to the genetics, it also seems a little less clear. So where it's very clear impact of loss of function of ANGPTL4 having a cardiovascular benefit. ANGPTL3, there are definitely some studies that show benefit, others that show it a little bit mixed, and maybe not even where there is a benefit, maybe not as large as ANGPTL4. So we do think that kind of really honing in on the lipoprotein lipase pathway explicitly through ANGPTL4 can be helpful.
Yeah, it's very interesting. Obviously the current therapy, monoclonal antibody for ANGPTL3 is for homozygous familial hypercholesterolemia.
Yes.
I think we're assuming that it's the endothelial lipase effect that increases remnant clearance in those patients
Right.
By uptake by remnant receptors and maybe some increased activity of the lipoprotein lipase. That drug doesn't work in people with familial chylomicronemia syndrome, which I would assume ANGPTL4 wouldn't either. You have a knocked out lipoprotein lipase. Am I correct? Or you haven't probably thought
We haven't tested it. We've thought a little bit about it. We certainly have no data to go either way, but our target is definitely ANGPTL4 and therefore lipoprotein lipase, so it may not have an impact in that population.
So those people with intact lipases obviously got a really brisk response. What was the total magnitude of triglyceride reduction, and what was the baseline triglycerides in your study?
Yeah, baseline triglycerides were just under 200. I think it was 193 mg per deciliter. And there we saw about a 53% reduction in trigs and also remnant cholesterol, which goes hand in hand. We did look at patients with baseline levels above 200 because I think everybody knows the higher you go, the larger magnitude, and there we saw more like a 64% reduction in triglycerides. So encouraging that you see that larger magnitude reduction as you go higher in triglycerides, which again we tend to see across triglyceride-lowering agents. So
sure, and even more encouraging, those are the people who have a lot of remnant lipoproteins.
Yes.
Which obviously we know are atherogenic, and to see those kind of major reductions in remnants is also very exciting. A lot more interest recently in remnant lipoproteins and their effect not only on coronary disease but peripheral vascular disease.
Yeah.
So when you're thinking about potentially in the future doing outcome trials, are there specific types of patients you'd be looking at, or were you going to? You're the development officer, so
Yeah.
I know that there's a lot of decisions to be made when you think about this. But were you thinking about maybe looking at pre-diabetics for progression to diabetes, or would you just be doing metabolic studies on those people in order not to have to study 10,000 people? And who would you be looking for specifically in terms of outcome trials?
Yeah, so our primary goal or focus is to go into a cardiovascular outcome study for cardiovascular risk reduction. Certainly that will include patients with diabetes or pre-diabetes, not necessarily exclusively, but metabolic dysfunction and diabetes, all of it goes hand in hand, right? You have the waist circumference and waist to hip ratio differences. You have elevated trigs, elevated remnant cholesterol, insulin resistance. All those things, as we all know, go hand in hand. So we would expect a high preponderance of people to have diabetes or to have pre-diabetes. What that means is you have an opportunity to look at things like insulin sensitivity or look at progression from pre-diabetes into diabetes. So that certainly wouldn't be a primary focus. The primary focus would be a pretty classic MACE reduction, but we would absolutely use that as an opportunity to be looking at potential benefits there. We don't have plans at this point to be looking at a different indication outside of the cardiovascular risk reduction at this point specifically. So we're not going to go after sort of, limiting conversion to diabetes or glucose resistance or any of those sorts of targets at this point.
The subgroup analysis.
Exactly, subgroup analyses. Now, again, we are doing some mechanistic work, so, data begets new decisions, and so you never know what data will bring. But right now, our focus is a pretty classic cardiovascular risk reduction.
Yeah. Have you thought about a severe hypertriglyceridemia trial?
So interesting. So I definitely, we think we'll reduce trigs substantially there. Our goal is still to go first into cardiovascular risk reduction, but this phase 2b study that we're going to run, we're actually doing a pretty broad range of triglycerides in there of 150 up to 880 or 10 millimole per liter, because we do want to explore the impacts in that more severe hypertriglyceridemic patient population. Also to somewhat explore where European guidelines are headed, right? So in the US, our guidelines are still very much at 500 mg per deciliter. Below that, you have cardiovascular risk, you address it. From those perspectives above that, you think about the risk of pancreatitis, right, and addressing there. But a lot of the European guidelines have started edging more towards that upper limit, maybe should be closer to 10 millimolar when you really start thinking about the risk of pancreatitis, as opposed to addressing cardiovascular risk in that patient population. So all of that to say, I'm not sure where our cuts would be in a cardiovascular outcome study, but we are using our phase 2 study as a chance to start exploring that.
Probably makes sense to stay below 880 so you don't get any FCS patients.
Exactly. Yeah.
Where you would anticipate you probably wouldn't see a lot of benefit, right, with a
But depending on the cause of
lipase,
it, but
right.
yes, if it's a knockout of lipoprotein lipase.
Well, this is really fascinating and exciting. I'm glad to hear the potential for not only atherosclerosis being looked at, because I think sometimes it takes lots of dollars and lots of patience to even consider one of those trials, and years go by. So congratulations on that. I thank you for my patient's sake. And also the idea that maybe we could see a reduction in risk of development of diabetes. So very fascinating. I'm going to give you the last word. Any final comments or summarize your program, your development program, and what you think is most exciting about about your medication. I don't know the name of it. I just know it's monoclonal
Well,
antibody.
we call it MAR-001, so
Okay.
for Maria and our lead asset. And I think we covered most of the data. Just to say, I think genetically, we feel like this is a strong genetically validated target with angiopoietin-like 4 and really compelling genetic data both for safety and for potential benefit, as we mentioned, in cardiovascular disease, but also in prevention of type 2 diabetes. The preclinical, the consistency across the genetic, the preclinical, and the clinical data, both for safety and for efficacy, is also really compelling. And we're just extremely excited to be moving into phase 2, the end of our phase 2 program, and very excited to head towards a cardiovascular outcome study. I've personally been in the space for a while. I was part of the Amarin team that ran the Reduce-It study, and we had a compelling therapy there, but certainly I think the trigs were part of it, but I don't think the trigs were the whole component, and I think this, the folks of MNLA know very well the bigger trig story, so I think it's also just exciting to be part of the science and trying to better understand what is the cardiovascular impact of elevated triglycerides and elevated remnant cholesterol, and how do we find therapies that can really help people with that residual risk. So very exciting time at Maria, and just so pleased to be here at NLA and have this opportunity to chat with you.
Thank you. It was a pleasure meeting, and thanks for sharing all that science with us. Wish you all the best of luck, and we'll all be excited to watch the progress of your development program.
Thank you. Very much excited too.